Hello guys and welcome to Meddy Simplified. So today we are going to talk about enzymes, a topic from biochemistry. So what are enzymes exactly? Enzymes are nothing but a biological catalyst. Enzymes are nothing but our biological catalyst. So your question will be there, what is catalyst? Catalyst is something which increases the rate of chemical reaction. So this word catalyst is something which increases the rate of reaction, increases the rate of chemical reaction. All right, anything which will increase the rate of chemical reaction. If I want to give you an example, it will be like a teacher-student relation. Sometimes what happens, even sometimes it happens with me, when we are studying from a textbook, we are not able to understand or grasp the topic very well as compared to when a teacher or someone on any platform is teaching you online or anyway, and then you understand it well. So that is what a teacher does, a role of catalyst in this thing. Like you will understand it, but it will not get lost in your memory, or it will take time. But when your teacher will explain you in front of you in any mode, then the understanding becomes very faster, right? So that is what a teacher-student relation is, an example of a catalyst. The teacher is acting as an enzyme for your memories for cracking the exams, right? So in this video we are more towards we will be more talking about the classification of enzymes, and by that means I will try and make you understand what are the different type of enzymes and how they are classified and what are their uses, right, and how they work exactly. So let's continue further. Next thing which we'll talk about is the classification of enzymes. All right. So basically there are two types of enzymes. One is simple enzymes, and second one is a complex enzyme. One is simple enzyme and second one is a complex enzyme. What is the basic difference between them? The simple one consists only protein part. So it is only proteinaceous kind of substance. So simple enzyme contains only protein. Whereas if you talk about the complex enzyme, they are something which has a protein part. Obviously, if the simple one is having a protein part, then why not a complex will have? So it has a protein part and a plus non-protein part. It has a protein plus non-protein part, right? So this protein part is called as something that is apoenzyme, whereas the non-protein part will be coenzyme or cofactor. So protein part is called as apoenzyme, whereas the non-proteinaceous part are coenzyme or cofactor, right? So this coenzyme or cofactors, the coenzymes are usually, I'll write usually, not always, but usually, coenzymes are usually vitamins, whereas cofactors are usually minerals, right? So this is the basic classification, simple and complex. Simple one consists only protein, whereas complex consists protein and non-protein. The protein part is called as apoenzyme, whereas the non-protein part is called as coenzyme or cofactor. Now there is a equation. I'll say that this apoenzyme along with this one coenzyme or cofactor, so I'll write coenzyme only. So apoenzyme plus coenzyme, they are together known as a term that is called as holoenzyme, right? So this is the equation. Apoenzyme, that is the protein part, plus the coenzyme is called as holoenzyme. So you can also say that complex enzymes are also known as holoenzyme, right? So what we should know more about this thing, that the apoenzyme, that is the protein part, is heat labile. If you have studied protein physical property, they get denaturate at a certain temperature. So similarly, this apoenzyme consists of protein parts. So it will break down into pieces when it is reached to a certain temperature. So they are heat labile. Whereas these coenzymes are heat stable. Coenzymes are heat stable. Alright, so these are the two things I wanted to tell you. Now recently in FMG exam once and once in the AIMS exam, there was a question that what is riboenzyme, right? So that brings us to the next topic, that is the non-proteinaceous enzyme. Some enzymes do not contain protein, so they are non-proteinaceous enzymes. So what are they? First is ribozyme, which I told you just now that there was a question on it, and second is abzyme. So if there is a question being asked on ribozyme, there is more chance to be asked about abzyme in the coming days. So ribozyme is RNA acting as a protein, acting as an enzyme. Whereas abzyme is antibodies. Abzyme is antibodies acting as enzyme. Antibodies acting as an enzyme. Right? So this was the basic thing which we should know about enzyme. Next, there was another classification called as IUB classification of enzyme, and that is the thing we should know, because the class of enzymes, it is divided into 6 types according to IUB system. So this six type of enzymes has been asked repeatedly in different exams. So we should know this classification, and then we'll end this video. It is a bit lengthier, not more than 10 or 12 minutes, but it is lengthier. So first we should write the heading. It is IUB system of classification for enzyme. So IUB system of enzyme classification. So in various notes I have seen many teachers give a mnemonic for this, that is OT Hill. But I'll tell you to not follow this mnemonic anytime, because the initials of this class of enzymes is not actually OT Hill, it is OTHLIL. So if you follow OT Hill, then it comes wrong. So just remember this thing, that it is OTHLIL. So you can, there is mnemonic operation theater has lights in light, so you can follow that one, but never OT Hill. So going further, so I'll write the classes this side first. So class 1, the IUB system has been divided into 6 classes. So class 1, class 2, class 3, class 4, class 5, and class 6. Alright, so these are the 6 classes. So OTHLIL. First is oxidoreductase. So class 1 is oxidoreductase. I'll give you the examples also and what they actually mean. I'll tell you in a while. First, let's write down the classes first. So class 1 is oxidoreductase. Class 2 is transferases. Class 3 is hydrolase. Class 4 is liases. Class 5 is isomerases. And last is ligase. Right? So oxidoreductase, there is a basic thing that if you add an oxygen or you remove a hydrogen, it is called as oxidation, and opposite is for reduction. If you add hydrogen or you remove oxygen, it is called as oxidation and reduction. Sorry. So once again I'll repeat it. If you add oxygen, it is called as oxidation. If you remove oxygen, it is called as reduction. If you add hydrogen, it is called as reduction. But if you remove hydrogen, it is called as oxidation. So both this reaction, oxidation and reduction, if they are occurring together in a single reaction, then that class using some enzyme, then that class of enzyme is called as oxidoreductase. So oxidoreductase are class 1. What is happening here? in oxidoreductase classes of enzyme, both oxidation and reduction are taking place together. Both oxidation and reduction take place together. Reactions together. Then next is the class 2, that is transferases. In transferases, what we are doing exactly, we transfer a functional group. So transfer of the functional group will occur when we use a transferase class of enzyme. So transferase is transfer of functional group. Transfer of functional group. Okay. Then next is class 3, that is hydrolase. Hydrolase, here we use a water molecule to break a bond. So hydrolase means breaking bond with help of water. And lyase means breaking. So lyase means breaking, but here we are not using any water molecule. So lyase means breaking bond without water. Then isomerases. In isomerases, isomerism will be seen, that is isomerization reactions is seen. What is this? I'll tell you in a while. Just write down. With the help of isomerases, isomerism takes place. Isomerization takes place. Alright. And then ligase. To bind anything is called as ligating anything, right? Ligating means binding. Ligating equals binding. So similarly, ligase enzyme is used to make a bond. So binding or joining two molecules. Is done using ligases. Right? So this is the IUB system of enzyme classification. Class 1 is oxidoreductase, class 2 is transferases, class 3 is hydrolase, class 4 is lyase, class 5 is isomerases, class 6 is ligases. Now I'll talk about each of them one by one with some example if possible. So first we'll talk about class 1. Class 1 was oxidoreductase. Right? So what happens in oxidoreductase? Oxidation and reduction reaction take place together. So suppose there is this reaction. A is having a H2, means hydrogen, plus we are adding it with B. So what will happen? In the A there will be a removal of hydrogen, whereas in B there will be a addition of hydrogen. Means in A there will be oxidation reaction and in B there will be a reduction reaction. So A will be added, will be removed, hydrogen will be removed from A, plus it will convert into BH2. So there the reduction reaction is taking place. So this is kind of example. The example of enzymes are, example of oxidoreductase are alcohol dehydrogenase, and next will be cytochrome oxidase. So these are two important examples of oxidoreductase to be remembered. Right? Next we'll talk about class 2. Class 2 were transferases. Class 2 was transferases. Here what was happening? Transfer of a functional group. So once again, suppose it was A with some X kind of functional group. So this is functional group plus B. So here what we'll do? We'll transfer the functional group from A to B with the help of any transferase enzyme. So here what will happen? A plus B with X, and this X is the same functional group. Right? So that this kind of transfer of functional group like phosphate, methyl, anything occurs with transferases. An example of such transferases are phosphorylase. Then we had hexokinase. We had transmethylase, transaldolase, like this. Many other examples are there. So this is transferase enzymes. Next we'll talk about hydrolases. In hydrolases, what we did, we break the bond with the help of oxygen. So next is class 3. Next is class 3 hydrolases. Class 3 hydrolases. Breaking the bond with the help of water. So suppose we have A and B. We added a water. If you look at the structure of water, it is something like this. Sorry. If you look at the structure of water, it is something like this. Okay. O is having one bond with one hydrogen and one bond with another hydrogen. Now using this water, we want to break this. We want to break this bond. Okay. So if for breaking this bond, this water will be divided into two parts. One side hydrogen, one with hydroxyl. OH को hydroxyl बोलते हैं. One with hydrogen, one part of hydroxyl. So what will happen? One group, that is A, will be added with a hydrogen, plus B will get a hydroxyl group. In this way, we'll break the bond. This bond is broken here. 1 second. This bond is broken with the help of this water. One H will go with A, suppose example, and one B will go with OH. So AH and BOH. This kind of breaking the bond with the help of water is called as hydrolases. Any enzyme with the name hydrolase, right? Suppose glucohydrolase, anything added with the hydrolase word is a something using water breaking the bond. Next is class 4. Next is class 4, that is lyase. Next is class 4, that is lyase. Here we break the bond without the help of water. So what will happen? Suppose we have A and B. We add with them with X and Y. So this X and Y will break down, and this A and B will break down. Means the X and Y together will break down the A and B, and they themselves will also break. And this kind of reaction is called as addition and elimination reaction. So what will happen? A will go with X and B will go with Y. So this kind of reaction is called as, this is also example of a kind of a addition and elimination reaction. Addition and elimination reaction. If you want any example of such enzyme, the lyase is having an example, that is aldolase. Aldolase. In glycolysis, you will see the 6 carbon eventually divided into two kind of thing, DHAP and glyceraldehyde 3-phosphate. So that breaking is with the help of aldolase. We have aldolase A and aldolase B. One is used in fructose metabolism, one is used in glucose metabolism. So that aldose enzymes are an example of lyase enzyme. They are breaking a 6 carbon into 3 carbon each. So that is lyase. Next we'll talk about the fifth one, that is our I. Class 5 is I, means isomerase. So there are few combination, two compounds which are having same molecular weight but different structure. So isomers, same molecular weight, but different structure. Right? Same molecular weight but different structure. Such kind of molecules are called as isomers of each other, and this isomerism takes place with the help of this thing, isomerase enzyme. Here what happens, there is an interconversion of functional group. There is a interconversion of functional group. Suppose we have a 6 carbon chain. Suppose we have a 3 carbon chain, and the OH group is on the second carbon. This is 1, 2, 3. Then there comes a isomerase enzyme. Isomerase. What will happen? It will change the functional group to third carbon. I'm giving just an example, right? So it will change the functional group to the third carbon. So what is happening? The molecular weight remains the same. The molecular weight of this enzyme will remain the same because it is having 3 carbon and 1 hydroxy group. Same is with this one, 3 carbon and 1 hydroxy group. but the structure is different. This is called as isomerism, and this isomerism take place with the help of enzyme isomerase. Then at last we have synthase. At last we have ligase. Ligase, I mean. So class 6th is ligase, means to bind. Ligase is to bind. So here we have two important example. One are the synthase group of enzyme, synthase, and another one is synthetase. Synthetase. So synthase are the one which are using no ATP to make a bond. No ATP used for binding two molecules. Whereas synthetase, the one with 2T, right? The one with 2T will use ATP. The one with 2T will use ATP. So synthetase uses ATP to bind two molecules. Right? So wherever in any reaction you will see the enzyme synthetase, right? So there will be utilization of ATP to bind two molecules. So that is about the ligases, and that was all about the 6 type of IUB classification of enzymes. Class 1 was oxidoreductase, class 2 was transferase, class 3 was hydrolase, class 3 was hydrolase, class 4 was ligase, class 5 is isomerase, and class 6 is ligase. And very quickly, one more thing I wanted to mention in this short video, that is factors affecting enzyme activity. Factors affecting enzyme activity. Right? So first is the enzyme concentration. Enzyme concentration. Second is our substrate concentration. Third factor is temperature, and last is pH. So this 4 thing can affect the enzyme's activity. So how do they affect? Enzyme concentration is directly proportional to the velocity of reaction. Directly proportional to the velocity of reaction. As you increase the enzyme, the rate of reaction will increase. Next is substrate concentration. It is also directly proportional to the velocity of reaction. As much substrate you will add, faster. More the substrate you add, faster will be the reaction. The graph will go on increasing. But about this temperature and pH, there is one thing that initially when you increase the temperature or pH, the velocity increases, but at a peak, there comes a peak when the velocity starts declining. So initially velocity increases, but after a peak it decreases. Right? So suppose if we draw a graph here about this. So this is on y-axis we have reaction of velocity. I'm really sorry for my diagram, but that's it. That's how I can draw. So suppose this is temperature and pH. So initially, as the temperature or pH increases, the reaction of velocity increases, and once it reaches a peak, it starts declining. Right? So this point, the maximum temperature, the point at which the temperature velocity is at maximum is called as Vmax. And the temperature at which we will reach the Vmax, and the temperature after which the graph will start declining is called as optimal temperature. And similarly, the pH at which the graph starts declining is called as optimal pH. So in exam there comes a question, what is the optimal temperature for the enzyme activity? So it is 35 to 40 degrees Celsius, and optimal pH for the enzyme activity is 5 to 9. Both of this will give this bell-shaped graph. This was also asked in one of the exams. So bell-shaped graph. Okay, so enzyme concentration and substrate concentration are directly proportional to the velocity of enzyme reaction, and temperature and pH initially they will increase. Once they reach the maximum velocity, the Vmax, it starts declining, and Vmax, the point at which it reaches the Vmax, is called as the optimal temperature or optimal pH. Optimal pH is 35 to 40 degrees Celsius, whereas optimal temperature is 35 to 40 degrees Celsius, whereas the optimal pH is 5 to 9. So thank you for watching this video. I hope this video was useful, and do not forget to like, share and subscribe my channel, Meddy Simplified. Thank you.